Hi everyone, my name is Lilja Quinn, and I'm a senior at Washington University in St. Louis. This summer, with the guidance of my mentors, Drs. Alta Howells, Srishti Kashyap, Broddrick, and Tori Hoehler, I have been working on a methanogen genome comparison project. So, what do I mean by that? Well, a methanogen is just an organism that produces methane. For this project, we focused on a single-celled organism in the genus Methanobacterium. And a genome is just the entire set of an organism's DNA. So, when I say Methanogen Genome Comparison Project, I just mean that we are comparing the DNA of different types of methane-producing organisms to see how they are related and what genes are and are not shared among different types. So, Methanobacterium species are chemoautotrophs, which means they get all the energy they need to survive by using chemicals floating around in their environment. Specifically, here you can see the chemical reaction that Methanobacterium typically use to make energy. They typically take in carbon dioxide and hydrogen to produce methane and water. So they live in many different environments, from animal stomachs to peat bogs, but they particularly like colonizing serpentinizing systems. These are water rock systems characterized by a specific set of chemical reactions that happen to produce molecular hydrogen, which, as the reaction above shows, is a key source of energy for Methanobacterium. However, these systems can pose some challenges to organisms trying to live in them. First, because of these chemical reactions, they have a very high pH, they're very basic, which can damage your cells. Second, while there is a lot of hydrogen in these systems, there is very little of the other quote-unquote food source for Methanobacterium dissolved in organic carbon compounds, so the other half of that equation. So where are these systems found? First, in various places on Earth. The system I focused on this summer was the Samyle ophiolite in Oman, and we believe on other bodies outside of Earth, such as Enceladus, the moon of Saturn. So why does NASA care about these little single-celled organisms? There are three main reasons. First, some researchers think that life on Earth might have started out in the form of chemoautotrophs in serpentinizing systems. Second, and relatedly, that means that other worlds, particularly Enceladus, that also have serpentinizing systems, have the potential to host similar life forms. And finally, because of their ability to take up CO2, a key greenhouse gas, methanogens might help us engineer carbon cycles on Earth or elsewhere. So, we wanted to understand, how does Samyle ophiolite Methanobacterium overcome the challenges of high pH and low dissolved inorganic carbon? Without getting too much into our methods because of time constraints, our main takeaway was that there were significant genetic differences between methanogens from serpentinizing systems and those from other environments. At the gene level, we saw differences in the enzyme, formylmethanofuran dehydrogenase, used to take up carbon. This enzyme catalyzes the first, most important, rate-limiting step of the conversion of carbon dioxide to methane. So seeing clear differences between serpentinizing and non-serpentinizing system methanogens implies that there is something truly different about how serpentinizing system methanogens perform these reactions. At the genome level, several functions related to pH regulation and carbon fixation are enriched overall in serpentinized system methanogens. We see genes with some of these functions more in these genomes than in our control group. And finally, over evolutionary time. This image is a subsection of our phylogenetic tree. These kinds of trees show the branching pattern of evolutionary relationships of a group of organisms over time. Here you can see that the Methanobacterium from the Samyle ophiolite, those marked with circles, form one group with a single common ancestor, right here. Because they share one common ancestor, this group is likely to share a suite of unique genetic changes. Overall, this brings us towards a clearer understanding of Methanobacterium. Our research points to a wide range of potential adaptations to carbon-limited, high-pH serpentinizing systems among Methanobacterium, leaving us a lot more to explore in the future. Thank you for watching, and thank you to my research advisors for guiding me in this process.